Well, good morning, everyone. Encourage you to grab a coffee. First thing Monday morning, here we are. My name's Rick Moss, and I'm the National Director for Strategic Partnerships at Duchenne Canada. Thank you for joining us for another in our annual series of webinars with industry updates. Duchenne Canada is committed to bringing the latest in Duchenne muscular dystrophy research and drug development to you, our Canadian Duchenne community. Before we get started this morning, just a couple of things to pass along, a few reminders. Use the chat feature to post comments and any questions that you might have. And then we will deal with those questions at the end of the session. We should have lots of time to answer any questions that you might have. And I think you'll find the presentation absolutely engaging as we go through this with our Dyne partners. For those of you who are new to our organization, just want to remind you that it was founded back in 1995 by John Davidson and Jesse Davidson. Known worldwide as Jesse's Journey. About 2 years ago, back in 2022, we rebranded as Defeat Duchenne Canada, a new name with the same vision, a future without Duchenne. Through our mission, we're focused on education and support for families living with Duchenne and Becker muscular dystrophy. We're advocating hard for access to treatments here in Canada and funding more than $17 million to research projects around the world. As mentioned earlier today, the webinar is one in a series of educational activities throughout the year to help educate and inform Duchenne and Becker families about activity here in Canada. And none of this work would be possible without support of some of our corporate partners, including Dyne, Pfizer, PepGen, and recently added Avidity. Well, now it's time to introduce today's session. Presenting today on behalf of Dyne is Dr. Ashish Dugar. He is the Chief Medical Officer for Dyne. Prior to joining Dyne, Dr. Dugar served as Vice President and Global Head of Medical Affairs at Sarepta Therapeutics. Previously, he spent 13 years at Pfizer in various roles, most recently serving as Vice President, Global Head of Clinical Development and Medical Affairs of the Biosimilars Business Unit. We are delighted to have you here today to provide an update on the initial clinical data, first released just back in January 3rd of this year, from the DELIVER trial of Dyne 251, a trial that's actually being conducted here in London, Ontario, as well at LHSC under the direction of Dr. Craig Campbell. For patients with Duchenne muscular dystrophy, amenable to exon 51 skipping. And now I'd like to turn things over to Dr. Dugar. Welcome.
Great, thank you, Rick. So as Rick said, my name is Ashish Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. First, I'd like to thank Rick and the entire Defeat Duchenne organization for the opportunity to present an exciting update on our Duchenne program today to the Duchenne community in Canada for the first time. Just before I begin, I'd like to introduce you to our new head of patient advocacy, Catherine Beaverson. As many of you know, Catherine has been part of the neuromuscular community for many years and brings tremendous experience and passion to this role. And for those of you who knew Molly White, who was previously in this role, please know that she's not going too far. She's taking on a new role here at Dyne. So I'm super excited for this addition to our team with Catherine's arrival, while continuing to be able to work very closely with Molly in her new role. And I have to thank Molly for building Dyne's key pillar, our delivering for patients core value, and connecting Dyne to this incredible community that we serve. And Catherine will be back on at the end of the talk for Q&A. Next slide, please. Next slide, please. Great. So I'd like to remind everyone that we'll be making forward-looking statements today, and you can refer to our website for more information. Next slide. At Dyne, it all starts with our mission, which is to deliver life-transforming therapies for people living with serious muscle diseases. Our mission is woven deeply into our core values, and it inspires our employees each and every day. Next slide, please. So the intentional design of the FORCE platform, which I have presented to this community in the past, is translating in the clinic from a safety as well as an exon skipping and dystrophin production perspective. And I'm so pleased to share those results a little bit later in the presentation. By way of reminder, the FORCE platform drives our efforts to develop and deliver muscle-targeted therapeutics. It is expressly designed to address one major issue in muscle diseases, which is to efficiently and reliably deliver antisense oligonucleotides like exon skipping PMOs to skeletal, cardiac, and smooth muscle, and target the genetic basis of disease. Our FORCE platform consists of three components. On the top left is the Fab, the antigen binding fragment or the antibody fragment, and it is the top portion of this Y-shaped figure, the full monoclonal antibody. So we took the top portion and designed it to bind tightly to something called the transferrin receptor 1 or TFR1, which is highly expressed on the surface of muscle cells. The second component is the linker, and we use a clinically validated linker, which we chose because of its demonstrated safety and efficacy in third-party approved products. And this linker enables precise connection of the Fab to the exon skipping PMO. And finally, we use an exon skipping PMO, which we rationally selected to target the genetic basis of disease. This is where we leverage the modularity of the FORCE platform. What I mean by that is that for nuclear targeting diseases such as Duchenne, we choose an antisense oligonucleotide payload. And in this case, we chose an exon skipping phosphorodiamidate morpholino oligomer, or a PMO, that targets the specific mutation in the DMD gene. And Dyne 251 is our investigational medicine that uses an exon skipping PMO designed to enable exon skipping of exon 51 to make shortened functional dystrophin. And the FORCE platform is designed with the goal of addressing current limitations of delivering antisense oligos to muscle tissue, to improve the delivery of the PMO to cardiac and skeletal muscle, including diaphragm, and also to allow for increased time between doses. Next slide, please. So the mechanism of FORCE is different from other approaches. We're essentially using the cell's natural biology to deliver our investigational medicine to muscle cells. Our Fab binds tightly to TFR1, as I said earlier. And again, the TFR1 is highly expressed across the surface of muscle cells, including skeletal and cardiac muscles. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter skeletal and cardiac muscle to target the genetic basis of Duchenne at the level of the nucleus. So the FORCE platform harnesses the natural mechanism of TFR1 mediated delivery to transport the PMO across the cell membrane, as you can see in the slide. And this is differentiating because we are not using any membrane destabilizing agents to deliver the investigational medicine into the cell or for the PMO to leave the endosome and travel to the nucleus. By targeting muscle specifically by using TFR1, we also have the potential for a wide therapeutic index, resulting in less frequent dosing and chronic administration. Next slide, please. So today, Dyne is well on its way towards achieving the promise of FORCE and delivering for patients. We've delivered exciting clinical proof of concept for our Duchenne program, and I'm pleased to share our initial readouts. Next slide, please. Prior to starting our Dyne 251 clinical development program, we proactively obtained expert feedback. We engaged key opinion leaders, or KOLs, from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. Importantly, we have gathered this feedback from across the globe. Now, KOLs in Canada, US, and outside of those regions, with experience running early and late stage clinical trials, have provided advice on our overall clinical development program. And this advice includes, but is not limited to, our overall clinical trial design, the patient population that we're studying, biomarker and functional endpoints, and key safety considerations. Importantly, they provided key learnings from numerous long-term natural history studies to help inform our trial design and eventually to contextualize our clinical trial results. It is absolutely clear that the KOLs believe dystrophin continues to be a key endpoint for this condition. We've also received advice directly from patients and caregivers, as well as global advocacy leaders such as PPMD, and from the Duchenne Community Advisory Board, comprising of advocacy leaders from several countries, including Canada. They have provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. Now, this input has been critical in ensuring a patient-centric design and has it also included, but is not limited to, the key considerations when families are choosing a clinical trial: review of and input to our informed consent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. And this advice has been invaluable and is directly reflected in the design of the trial. Next slide, please. So before we go into detail on the data, I want to provide a brief overview of the DELIVER study design. It's a phase 1/2 global trial that we have designed to be potentially registration. The trial is enrolling ambulant and non-ambulant males with Duchenne who are ages 4 to 16 and have mutations amenable to exon 51 skipping therapy. Patients who have been previously treated with exon skipping therapy can enroll and deliver after a 12-week washout period. The primary endpoints are safety and tolerability and change from baseline in dystrophin levels as measured by a technique called Western blot. Secondary endpoints include measures of muscle function like NSAA and time function test, as well as exon skipping. We also have wearables to allow collection of data such as stride velocity. Next slide, please. So the DELIVER trial consists of 4 parts. There's a 6-week screening period, a 24-week multiple ascending dose or MAD, randomized placebo-controlled period, a 24-week open label extension, and a 96-week long-term extension. In the MAD placebo-controlled portion of the DELIVER trial, participants are randomized to receive Dyne 251 or placebo every 4 weeks, and then all participants receive Dyne 251 every 4 weeks after the MAD portion. Again, we have a number of outcomes measures to evaluate function and quality of life. And for details on inclusion and exclusion criteria, you can visit our ct.gov page for more information, and the identifier is at the bottom of this slide. Also, we do have sites that you can see on our ct.gov page, and we do have sites in Canada, as Rick mentioned, with Dr. Craig Campbell at Children's Hospital, London Health Sciences Centre, as well as Dr. Hugh McMillan at Children's Hospital of Eastern Ontario. Next slide, please. Very briefly, this is the trial design. I'll just take a second to go through this. So as I mentioned, in the MAD placebo-controlled portion of the DELIVER trial, participants are randomized to receive Dyne 251 or placebo every 4 weeks. In the US, we started at 0.7 milligrams per kilogram of the PMO equivalent dose, and you can see that on the bottom bar in blue. And outside of the US, including Canada, we initiated the trial at 5 milligrams per kilogram PMO equivalent dose. Open biopsies are taken at baseline at 6 months and only in the 2.8 mg per kg cohort and higher. In the first 4 cohorts, we randomize at a ratio of 2 to 1, meaning for every 2 individuals on Dyne 251, 1 individual receives placebo. And starting at the 10 mg per kg dose, individuals are randomized 3 to 1. And at 10 mg per kg and higher, these will be our dose optimization cohorts. It's truly remarkable to see the enrollment progress. 40 participants have enrolled through the 20 mg per kg cohort. Next slide, please. Now I'll review the initial data results. First, we're very pleased with the favorable safety profile for Dyne 251 that we've observed in DELIVER as of the data cutoff date. This is very differentiating from others in the field and speaks to the importance of each component of the FORCE platform, as I mentioned earlier. The Fab, so a portion of the antibody, not the entire antibody, the clinically validated linker, and the choice of the PMO. Overall, the safety profile of Dyne 251 has been favorable through the data cutoff date. The safety data is based on 37 participants dosed through the 20 mg per kg cohort of the MAD portion of DELIVER. Most treatment emergent adverse events were mild to moderate in severity, with one serious treatment emergent adverse event that was deemed unrelated to study drug per the study investigator, and that was dehydration due to gastroenteritis. There have been no reports of anemia, thrombocytopenia, kidney injury, and also no clinically meaningful changes in electrolytes, including magnesium, have been observed. The safety profile has supported dose escalation through 20 mg per kg cohort, and approximately 275 doses have been administered in DELIVER as of the data cutoff date. Next slide, please. So turning to dystrophin expression as measured by Western blot, which is the co-primary endpoint of DELIVER along with safety and tolerability, we'll go through these results now. So the gray bar represents patients in the placebo-treated group in DELIVER, and the blue bar represents the Dyne 251 treated patients in DELIVER. Patients treated with Dyne 251 had a mean absolute dystrophin level of 0.88% of normal and a 0.28% change from baseline at 6 months, while the placebo-treated group was largely unchanged. On the right side of the slide, you can see data published in the Journal of Neuromuscular Disorders, where eteplirsen, the current standard of care in the US, showed a mean absolute level of 0.3% of normal and a 0.06% change from baseline at 6 months. In looking at these studies side by side, Dyne 251 had a 2.5-fold higher dystrophin expression at 6 months than eteplirsen, with a 24-fold lower total PMO dose administered 4 times less frequently. Now, our goal for this initial read was to be at least as good as standard of care at 6 months with a very different dosing paradigm, that is once a month, which is meaningful for patients. So these data are compelling, and we believe we're on track to achieve higher levels of dystrophin in our dose optimization cohorts, the 10, 20, and 40 milligrams per kilogram PMO equivalent dose cohorts, where we would hope to see 5 to 10% dystrophin production. We know from the work of others in the field that dystrophin builds over time, and others have seen a doubling effect out to 96 weeks. And we believe a 10 to 20% dystrophin production would be able to significantly alter the trajectory of disease for patients. Next slide, please. Next, we looked at percent dystrophin positive fibers, or PDPF. This measure is important because quantifying localization of dystrophin to the cell membrane helps provide confidence that there should be functional benefit. Again, the gray and blue bars represent placebo and Dyne 251 treated groups respectively in DELIVER. And you can see that Dyne 251 demonstrated 22.2% mean level of dystrophin positive fibers and a 19.8% change from baseline at 6 months. In the eteplirsen trial, published again in Journal of Neuromuscular Disorders, eteplirsen showed a 19.6% mean level of PDPF and a 10.7% change from baseline at 6 months. Next slide, please. So to briefly summarize these compelling DELIVER data, at this initial 5 mg per kg PMO equivalent dose delivered every 4 weeks, Dyne 251 showed a compelling profile versus published data from eteplirsen standard of care at 6 months. And that includes a 2.5-fold higher dystrophin expression with 24-fold lower PMO dose administered 4 times less frequently, as well as a 2-fold higher change from baseline PDPF versus what was seen in the published eteplirsen study. We've also seen a favorable safety profile to date in dosing up to the 20 mg per kg PMO equivalent cohort dosed every 4 weeks. That cohort is now fully enrolled with 40 patients total enrolled in DELIVER with approximately 275 doses administered. So we're really pleased with these initial results. And in terms of next steps, we will continue to optimize the dose and dose regimen in 2024. That is a key part of what we will be doing in the DELIVER study this year. And also, we will have our next clinical update from DELIVER in the second half of this year. And I look forward to coming back to this community to give you an update later this year. Next slide, please. So we believe the data set from DELIVER illustrate the potential of the FORCE platform in developing next generation exon skipping therapies and building a global DMD franchise. Historically, muscle delivery has been the challenge, and these initial data of Dyne 251 demonstrated targeted delivery to muscle with the potential for a wide therapeutic index where other approaches have struggled. And Dyne is committed to developing a DMD portfolio leading with Dyne 251, and we've identified PMOs for mutations amenable for skipping exons 53, 45, and 44. Next slide, please. And finally, we have tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We are working urgently to deliver for the community, including families and healthcare providers, to bring a potentially transformative treatment forward. Thank you to the families for being generous with your thoughts, advice, and for the sacrifices that you continue to make to advance science and medicines. So on behalf of Dyne, I'd like to thank you all for your time and attention to listen to this presentation. Thank you again to Defeat Duchenne for having us. And Catherine and I are happy to take any questions.
Dr. Dugar, thank you very much. And Catherine, welcome. There's a couple of things that have come up. And one of the questions was, you mentioned a 10 to 20% increase in the production of dystrophin would significantly alter the disease. Can you just, in lay terms, explain what would that mean for their son?
Sure.
If in fact that was achieved? And I guess, is that a realistic expectation out of this project?
Sure. So let me actually maybe back up a little bit, and then I'll get to your question. So what we've seen from other exon skipping programs is that over time, dystrophin accumulates. And so, as I mentioned, with other data that's out there in patients, you can even see a doubling of effect out to 96 weeks. So we do know dystrophin accumulates over time, and it just takes 1.5 years or 2 years to sort of reach steady state. So within our trial, we are actually seeing dystrophin levels that are matching our modeling projections. So we're encouraged that, as we think about what we might possibly achieve at 5, 10, 20, 40 milligrams per kilogram based on our modeling of dystrophin increase with those dose levels, we're on track. So we are hoping, as I said, that as we get to that 10, 20, 40 mg per kg, we hope to see in a 6-month period that 5 to 10 dystrophin production. And then if indeed we follow the trajectory seen with current compounds and a doubling of effect over 1.5 years or 2 years, that could reach as high as 10 to 20%. So in terms of what this really means for families and for individuals with Duchenne, current treatments just haven't gotten to those levels, so no one can say definitively what would be the case. That said, there is published literature. Specifically, there is a lab out of France, most notably from Dr. Amthor, Professor Amthor. And in that particular paper that was published out of that lab, his group looked at patients that had different levels of dystrophin. So just within, not on treatment, but just, patients who had different levels of dystrophin. And one can see in this cross-sectional study that patients who had 5% or more dystrophin had a different phenotype, sort of a different rate of progression towards loss of ambulation, et cetera, versus those who had less than 5% dystrophin. So there is data out there, clinical data out there, that indicates if you get to those levels of 5 and above, that it actually should lead to a different kind of trajectory of disease in terms of mortality and loss of ambulation and the like.
Great. Thanks, Dr. Dugar. We have just a comment really from one of the parents. Her son is in the Dyne study here in London, going on dose 11. She just wanted to share that her son has been doing his research and is finally optimistic. He feels some slight changes. So they thank you for checking in with these updates. It's great.
Our pleasure.
I also wanted to find out a little bit more, and perhaps, Catherine, this is a question for you, the engagement of the community in this exercise. Not everyone is doing that, unfortunately. I think it's a great initiative. What was the response from the families when you reached out to them in that engagement process?
So I'll confess that I was not part of the study looking at the involvement, but given my experience over the last decade, I think it starts with the appreciation that families and leaders in this community are really experts in their lived experience. They have learned over decades of experience in clinical research how to engage in the scientific process. And so I think, it's a win-win when companies can work with the patient community to tap into that expertise to understand what motivates people to participate in clinical research, what's the risk benefit profile, how can they engage in clinical trials to the maximum of their lived experience, what are elements that are burdensome, what are elements that are feasible, and so kind of understanding how people come into research and what keeps them in a clinical trial only improves the process for both.
That's great. Yeah, and very true.
And I'll just add that, the families that were specifically involved in the design of the trial expressed all of the sentiments that Catherine just mentioned.
That's great. And your consultation with them will continue as the trial continues, I'm sure.
100%.
Great. We have a couple of questions coming in now. The first one is, can my son stay on vamorolone while participating in this study?
Yeah, so it's a great question. So we are going to so for those jurisdictions where vamorolone receives marketing authorization, they will be allowed to be on vamorolone. So that is the plan. For those jurisdictions where it is not yet approved or does not yet have marketing authorization, it is still considered an investigational medicine. And so there are restrictions on the use of other investigational medicines while doing a study for this particular investigational medicine. And that can be found on the ct.gov page as well.
The next question, for the 20 milligram dosing, when would the preliminary timeline of results be?
So we haven't given specific guidance on when we will be releasing results on a per cohort basis. And so, my apologies, I'm not in a position to do that today. That said, there will be additional clinical data released in the second half of this year, but we just haven't given guidance as to exactly what data or which cohorts.
Okay. And a couple of other questions from the same family here. Based on these initial results, is the company working on second generation platform and learning from the outcomes of these trials?
Oh, yeah, 100%. I'd love to go into a ton of detail, and I know we don't have the time. But first of all, even with respect to the current technology we have, we still are doing more work, as I mentioned, to optimize the dose. And you may have also heard me say sort of dose regimen optimization. So based on the data that we've accumulated so far in the preclinical and clinical space, we believe there's also the opportunity for less frequent dosing than every 4 weeks. So we're really excited about really learning this year and optimizing that dose and dose regimen for delivered Dyne 251. We continue to look at various technologies to even go beyond in terms of being able to have a platform that can deliver other platforms that can even more robustly deliver antisense oligonucleotides. So we're definitely not sitting back and just waiting for this platform and see what we can do, but we continue to work on next gen platforms because we know that the community needs as many options and shots on goal as possible. So yeah, we're definitely continuing to do more work with this platform and next generation platforms.
And a final question on this group. Could you tell about the French doctor's name again, where the clinical data is available?
Sure. So it's Professor Helge, Helge. I hope I'm spelling that right. And Amthor, Professor Amthor, Amthor. So you'll be able to pull up that manuscript, and I'm happy to send it over to you, Rick, as well,
Sure.
if you'd like.
Great, and we can share this with the family. That'd be great.
No problem.
Good. The other question, of course, that we've had is the how transferable is this learning to other exon skipping treatments?
Great question. So I don't know that I mentioned this, but our FORCE platform utilizes the same antibody, the same Fab, targeted to the TFR1 receptor, and the same linker. So this is designed to be modular in nature so that, within Duchenne, we can use the same Fab and linker and attach a PMO that's appropriate for skipping exon 53, a PMO that is appropriate for skipping exon 45, for example. So what we learn from this is directly applicable to developing treatments for other mutations for Duchenne. So we're going to learn a great deal, not just from what this does for 51 skip amenable patients, but also for others from a safety, tolerability, and efficacy perspective. So it's directly applicable.
Well, that is great. That brings us to the end of the questions. Is there anything else, Dr. Dugar or Catherine, that you want to add before we close?
Catherine, anything else before we end?
No, just thank you for allowing us to present today. And as this last slide says, if there's any questions, the community is free to reach out to me, and please feel free to extend the invitation beyond the participants in this webinar. Thanks, Rick.
Catherine, thank you so much. Dr. Dugar, always
Thank
a pleasure. And to Molly and Tiffany, thank you to them and best wishes to the team. Keep up the great work. We have a few things that we're going to cover off, some housekeeping items here for the rest of the webinar, and then we'll close out. But thank you both for participating. We'll talk again soon. It sounds like the second half of this year.
Absolutely.
Best of luck.
Thank you so much, Rick.
Thanks for having us.
Our
Bye-bye.
pleasure. Also wanted to share with our audience this morning our research grant announcement that was just 2 weeks ago. We were excited to share that we'll be granting $880,320 to 5 promising research projects this year, bringing our grand total over our 29-year history to almost $18 million. Also, we have a busy month in February, along with this webinar. Rare Disease Day, of course, is coming up. Defeat Duchenne is launching a self-submission storytelling platform. We invite you to participate, and you can see the details as to how to do so on the screen. You'll be seeing more of that and hearing more about that in our e-blasts, on our socials, and also on our website. And of course, the big annual event that we hold, the Walk to Defeat Duchenne, is set for Sunday, May 26th, just announced. Please gather friends and family to participate by completing the 33K Founders Challenge, or choose your own distances, whatever you like to do. But that date again, Sunday, May 26th, we'd love to have you join us virtually for that one, and along with hundreds of others across the country. And a reminder of some of the services we do offer, the emergency care resources that are available. Go to our website to see how you can acquire those. And if you have any additional questions, please feel free to reach out to us, and we'll be happy to try to answer them for you. Also, clinical trials. We heard of the trials that are going on in Canada with Dyne at CHIO in Ottawa, at LHSC here in London, Ontario. Just some of the clinical trial finder tool locations that you'll see with our tool. It is on our website, of course, and you can look and see where trials that perhaps for your son are open and available. We have a lot of other items going on here, of course, throughout the year, and we'll keep you posted. This is the first in our series of webinars. We'll not only be doing educational webinars, but also clinical webinars and pharma updates as well. The activity in the Duchenne world is accelerating at a rapid rate. We're seeing more and more research coming out and results coming out. And that's where our role kicks in to try to keep you abreast of everything that's happening on the Duchenne front. If you have anything you'd like to see done in a webinar from an educational standpoint, questions about transition, whatever they happen to be, we'll look at that and try to set something up with the experts that we're associated with and get you that information. I just want to thank everybody again, remind you of our newsletter, to stay in touch with us and keep communication lines open. Sign up for our new newsletter if you haven't done so already at simplydefeatduchenne.ca. Thanks again to everyone this morning who joined us. Thanks to the Dyne team. We look forward to doing this again soon. We'll keep you posted. Watch our website and our e-blasts and socials for more information. Thanks, everyone. Enjoy the rest of your day.